Central metabolism, reducing electron carriers via glucose oxidation. We're back to explore how we load or reduce electron carriers. It's a big topic. Glycolysis, pyruvate oxidation, and the citric acid cycle are collectively the backbone of central metabolism. In each process, NAD+ gains electrons, reducing it to NADH. Those electrons come from oxidizing glucose, which unfolds over roughly 20 steps. Breaking glucose oxidation into so many steps slows its energy release. If it all happened at once, it would be like a glucose rocket or a raging campfire, rapid energy release that would damage cells. We're about to dive deeper into these processes, so keep three principles in mind. First, conservation of matter. Glucose's 6 carbons can't just vanish, so we'll track each one, keeping us oriented through these complex pathways. Second, redox pairings. Electrons need a donor and an acceptor, like trick-or-treating, where you can't get candy unless someone gives it. NAD+ is reduced only when a molecule is oxidized, often carbon from glucose to CO2, providing electrons to make NADH. So each CO2 released yields one NADH. Central metabolism produces one NADH for each of the 6 carbons released as CO2, but 4 additional NADH and 2 FADH2 also form from oxidations that don't produce CO2. Third, enzymatic catalysts. Enzymes lower the activation energy of each step, allowing reactions to proceed at biologically useful rates. And they serve as key targets, allowing the cell to adjust how fast reactions run. Though enzymes won't be shown in this video, they're responsible for everything we'll talk about. Let's start with the lone glucose. 6 carbons, each more beautiful than the last. Glycolysis splits it in half, investing 2 ATP up front and producing 4 ATP by substrate-level phosphorylation. That's 2 per 3-carbon piece, for a net of 2 ATP, the maximum yield for organisms lacking an electron transport chain like lactobacillus. Each 3-carbon fragment also reduces an NAD+ to NADH, so glycolysis nets 2 NADH as well, and ends with 2 3-carbon pyruvates. So the total products of glycolysis are 2 net ATP, 2 NADH, and 2 molecules of pyruvate. We'll track just one of the pyruvates, but remember everything happens twice from here on. The transition reaction, also known as pyruvate oxidation. Here, 1 carbon is released as CO2. Its oxidation reduces NAD+ to NADH, a CoA tag is added, and we yield acetyl-CoA that's ready for the citric acid cycle. Originally, Hans Krebs named it the citric acid cycle after its first intermediate. It also became known as the TCA, or tricarboxylic acid cycle, due to the citric acid's 3 carboxyl groups. After Krebs won the Nobel Prize, his colleagues renamed it the Krebs cycle. To everyone who felt the urge to invent a fresh name, thanks a lot. Now biology has even more redundant vocabulary. The citric acid cycle's job is to oxidize the last 2 carbons of acetyl-CoA to CO2, producing NADH. To start that process, those 2 carbons are passed to a 4-carbon molecule called oxaloacetate. You don't need to know the name, but I'll use it for clarity. Combining them yields a 6-carbon molecule called citric acid, hence the cycle's name. From there, the carbons are oxidized to CO2 one at a time, generating 2 NADH and a 4-carbon intermediate. When that intermediate is oxidized back into oxaloacetate, an ATP is produced by substrate-level phosphorylation, and FAD and NAD+ are reduced into FADH2, and another NADH. Now, oxaloacetate is ready for another round. We've now loaded many electron carriers, which will travel to the ETC, drop off their cargo, and return to the cytoplasm. And that, in a nutshell, is how glucose from our food becomes the ATP that powers every cellular task needed to survive. By running glucose through glycolysis, pyruvate oxidation, and the citric acid cycle, our central metabolism reduces electron carriers into NADH and FADH2, which then feed electrons into the electron transport chain. Passing those electrons generates a proton gradient, which flows through and drives ATP synthase. In other words, from sugar to NADH to proton gradient to ATP. Every step is chained together so our cells can do the work of life. Next time you eat, take a moment to appreciate this carefully choreographed series of reactions that keep your cells alive.